Hello, this is Vahidan and welcome to my YouTube channel. Today we are going to discuss about the biomolecules. Biomolecules. Yesterday I have given an introductory part of biomolecules. Today we are going to discuss in detail about the carbohydrates, which is the first biomacromolecule we need to discuss. Yesterday I have defined certain biomolecules, which are actually also known as biomacromolecules, as they have subunit, as they have monomeric unit. We know that biomolecules are protein, lipids, carbohydrate, and nucleic acid. Enzyme has been included in your syllabus, but it should be under the topic of proteins as they are protein in nature. But we are going to discuss in detail. So today our lecture is all about the carbohydrate, and in addition to all these biomolecules, we need to discuss the nucleic acid, as nucleus is part of your syllabus. So nucleic acid subunit or monomeric unit is nucleotide. Moving on to the first slide of this lecture. So I am going to define about what are biomolecules. Basically, they are essential organomolecules which regulate the physiology of the cell. It's a footprint, or it's a soldier, or it's something that is necessary for the survival of living organism. So I have these are non-living molecules are the actual foot soldiers or the battle of sustenance of life. Actually, without them, our life would not sustain. We know that our body is made up of all about proteins, carbohydrates, lipids, fats, and nucleic acids, DNA. So, if we neglect these molecules, life we can't be called a living organisms. Okay, next, they can be from small range as primary metabolites and secondary metabolites, and to the large macromolecules like proteins, nucleic acid, carbohydrates, and lipids. Okay, moving on to the next slide. Okay, these are the structures of the biomolecules. Lipids play a vital role in the formation of cell membrane, and I have discussed in detail that plasma membrane is a fluid mosaic model and consists of phospholipid molecules. Next, nucleic acid. Nucleic acids, I have told you that nucleic acids are present in the nucleus of cell as they contain DNA and RNA. DNA is deoxyribonucleic acid. Then moving on to the next structure of I have drawn a structure of carbohydrates. It's a polysaccharide structure where polysaccharide molecules are combined together to form the carbohydrates. Are the examples of carbohydrates. Next, the proteins. We are going to discuss each and every in different tutorial. Today our topic is all about the carbohydrates. So moving on to the next slide. Okay, here I have defined what are carbohydrates. I told you in the previous lecture that the general formula of carbohydrates is CN bracket H2OM. That is actually the general formula of carbohydrates. But I need to define that chemically they are basically polyhydroxyl group and polyketone groups. So in layman terms they are actually sugars which taste sweet in it. But one thing I want to tell you that basically polysaccharides, as we move as the number of carbon atoms, as the number of monomeric units increases from the monosaccharide to the disaccharide and to the finally polysaccharide, basically the sweetness of the carbohydrate is reduced. So I am going to define here carbohydrates are chemically defined as polyhydroxyl aldehyde or ketone group, polyhydroxy aldehyde and ketone group. This is the structure of basically this is a chemical formula or a functional group of aldehyde, and ketone is actually bonded to oxygen atom with other two variable group. But this is actually a chain of uh, other molecules. But mostly, I am going to tell you that hydroxyl groups lie at the end of the molecule at terminals, while ketone group lie within the basically molecule. We are going to deal with such example in the further slides. Okay, depending on the number of constituting sugar units upon hydrolysis, they are classified as monosaccharide. When monomeric unit, monosaccharides. Two monomeric units, it's disaccharide. And basically, I have classified disaccharides into polysaccharides, but disaccharides only contain two monomeric units, and polysaccharides start from the 3 to 7. And further on, in the increase of monomeric unit, glucose is one monomeric unit. Okay, glucose is a one monomeric unit. When two glucose combine together, they form a disaccharide. It's a two monomeric unit. When three glucose molecules combine together, it forms a polysaccharide. When many monomeric units such as n number, thousands and hundreds of number combine together, they form polysaccharides. When these polysaccharides are attached to attach to certain other biomolecule or monomer such as a fatty acid or protein. They form basically oligo basically complex oligosaccharide. So they have multiple functions. They are important and most important dietary source. This is one of the reasons I said they are the instant source of energy for the human, for every organism. They are structurally very important for many as they form the major structural components such as cellulose is an important structural fiber for plant. As we know, we have discussed in the cell biology topic of basis of life. There I have told you that plant cell contains cell wall, and cell wall is made up of cellulose. So there they play a role in the structural integrity of cell wall, and that give him the strength to the cell wall, and that's why the plants have rigid cell wall. Moving on to the next slide. Okay, so I have in detail. I am going to discuss about the first detail of each category of carbohydrates. First, we are going to discuss about the monosaccharides. Monosaccharides are usually have general formula CH2ON. But in case of polysaccharides, this is the same I have told you. It can be written for all it can be written for all it can be written for all polysaccharides. So all carbohydrates. This is the general formula for all carbohydrates, but specifically they have derived the general formula for this one. Okay, monosaccharides usually have general formula CH2O, and where N can be 3, 4, 5, 6, and have 2 or more hydroxyl groups. Okay, it may have 2 and more. Like C, 3 carbon. This is aldehyde group, as I have brought above. C double bond to OH at terminal end. Aldehyde group will be at terminal end, while ketone group will be within the basically within the molecule. And carbon is bonded to 2 other carbons. Carbon is bonded to 2 other carbons. Carbon is bonded to 2 other carbons. In case of basically ketosis. Okay, they are either contain aldehyde group, so they are called aldoses. When they contain ketone group, they are called ketosis. So I am going to tell you one thing. Aldoses 3 carbon is also called triosis, and specifically gassy-aldehyde. When this is the same 3 carbon atoms here in aldose sugar, same 3 carbon atoms are here, but the bonding of oxygen to the carbon at the terminal and converted to aldoses, but the bonding of carbon in the middle section convert into ketose sugar. So this is the difference between ketone and aldehyde group. So the same sugar with aldehyde group is called glyceraldehyde, and the same number of carbon atoms containing that sugar also is called dihydroxyacetone, because the carbon atom has changes position, so the name of that molecule has changed. So 5 carbon sugar pentoses. In case of in case of aldose, it's a ribose sugar. In case of ketones, it's a ribulose sugar, which is actually found in the basically pathway of photosynthesis or something like that. Okay, aldose 6 sugar is called glucose, and ketose 6 carbon sugar is called fructose. These are all monosaccharides. And in the I have told you that in the CSS examination 2020, there was question what are carbohydrates, classify them, and give the example. So these are the examples of monosaccharides. Okay, they either contain aldehyde group, they may contain, we have given the example glyceraldehyde, ribose, glucose, or they may either contain dihydroxyacetone, which example is dihydroxyacetone, ribulose, fructose. If it is difficult in learning these names, you can just give the example of glucose and fructose, which is a 6 carbon sugar. Okay, moving on to okay, these basically I am going to discuss about the isomers. These are basically isomers of monosaccharides. So many monosaccharides differ only in the special arrangement of atom, that is they are isomers. When the arrangement of atom is changed, so basically the form of sugar is changed. But they are all examples of monosaccharide. For example, glucose, galactose, and mannose have the same formula. What is the formula of glucose? C6H12O6. Okay. So what is the formula of galactose? Yesterday I have told you it's also the same. What is the formula of basically mannose? It's also same. But what is the difference? That at basically carbon number 1, 2, 3, 4. That's 4 carbon has a hydrogen hydroxy group at the upper end and hydrogen group at the lower end. So that's galactose. When this spatial arrangement is basically modified, this is actual arrangement is this one. This is a glucose molecule at the 4 carbon. We can see that position of basically what we call hydroxyl group is downward and hydrogen is upward. So that position is changed in galactose, and the position of other carbon where hydroxyl and hydrogen change is mannose. So this the arrangement of hydroxyl group and hydrogen at different atoms results in the formation of isomers of glucose. So these are these small differences make only minor changes in the chemical properties of sugar, but the differences are recognized by enzymes and other proteins and therefore have a major biological effect. So, what I'm trying to say here is that these all are monosaccharides, these are all sugars, but the difference is that they would also provide energy, but there is that they have a different aspect to play. They have different role to play in our body. Each of the carbohydrate monosaccharide I have written here, which is also one of each other, has basically different enzyme for its digestion. So that is the reason that a single change in the structure of these monosaccharides results in a different biological effect, and they also have different biological pathway through where they release energy. Moving on to the next slide. Okay, glucose may form a ring structure, or they may be open structure. In aqueous solution, the aldehyde and ketone group of sugar molecule tends to get in a hydrogen group of the same molecule, thereby closing the molecule into ring. So, what I am trying to say here is that basically in water or in aqueous solution, in water basically that is present in aqua, when glucose is present, it tend to shape its structure in a ring formation. So as we know that this is basically glucose and this is ribose. They have basically what is that? They have changed into ring structure. What happened to that? That is that oxygen has been basically linking them. So this is in aqueous solution that we don't know. You can see that that's the aldehyde group, and which has changed its conformation in glucose, and that is also the aldehyde group which has also changed in conformation in when placed in aqueous solution. So in aqueous solution they tend to form ring formation occurs, and their hydroxyl group, polyhydroxyl group of aldehyde and ketone reacts. It does not react, but it's basically changes help in the changing of conformation. So moving on to the next slide. Okay, so there is one thing I want to tell you, that alpha and beta linkages are very important. These are the linkages which result in the formation of different polysaccharides. When glucose is linked through alpha linkage, it has different polysaccharides resulting. When it is linked to beta linkage, it has different basically aspect. The hydroxyl group of carbon atom carries the aldehyde or ketone group, can rapidly change from one position to another. These two positions are called as alpha and beta. When basically at second carbon, hydroxyl group is at the upper side of the ring, near to the aldehyde group oxygen. It's a beta hydroxyl group. When hydroxyl group is below, at the second carbon is below or is away from that of the oxygen, so it's a alpha hydroxyl group. As soon as one sugar is linked to another, the alpha or beta form is frozen. Okay, what I mean to say? I mean to say the same thing, that in formation of polysaccharide, these linkages, if it is present in this conformation, it would be frozen in the polysaccharide in that conformation, and that would result in a different polysaccharide. If hydroxyl group is at the downwards end and that reacts in a solution that forms the polysaccharide, that will form a different polysaccharide. So change of spatial arrangement of a hydroxyl group has a very important role in the formation of polysaccharides. That's why alpha and beta linkages are important. So moving on to the next slide. Okay, these are the sugar derivatives or monosaccharide derivatives. We know that I have told you about the complex oligosaccharide, which I am going to discuss later on. These monomeric units may link to other basically groups resulting in basically what we call sugar derivatives. This is basically an example of one glucose where the COOH, which is acidic group, carboxylic acid group is attached, so it forms glucuronic acid. So when amino group is attached, it's from glucoamine. So when some N-acetyl group is attached, so it's from N-acetyl glucoamine amine group. So these are the sugar derivatives. There are certain examples. If you want to learn, go ahead. I will fetch you more marks, but there is no need to learn. I just need to realize that as in plasma membrane, various carbohydrates are by the Golgi operators are linked to the proteins. So this is how I am going to define you these things. Moving on to the next slide, where we are going to discuss about the disaccharides. When two monomeric unit or when two monomeric unit of monosaccharides get together, they form disaccharide. Okay, there is one thing I want to discuss. There are reducing sugar and there are non-reducing sugar. Most of the time in a PTSC, FPSC, or competitive examination, there is a question: what is the example of non-reducing sugar? Remember that is always sucrose. That is always sucrose. So, I'm giving you the example of disaccharides. The carbon that carries aldehyde or ketone group can react with any hydroxyl group on the second sugar molecule to form a disaccharide. It means that two monomeric unit of monosaccharides are linked together. This is alpha glucose, this is the beta fructose. When they react together, water is removed from them. That results in release of water from this, and that results in the formation of sucrose, which is actually the non-reducing sugar. Okay, so maltose glucose plus glucose resulting in the formation of maltose. When two glucose molecules react together, they form maltose. When one galactose and one glucose react together, they form lactose. And when glucose and fructose react together, they form sucrose. We know that fructose and glucose are actually the isomer of each other. They have different special arrangements of hydroxyl group. But this is how when two monomeric units of monosaccharide react together, they form basically form disaccharide with the release of water. And actually that is also called hydrolysis. That is no, that's not hydrolysis. That is actually the removal of water from there. Okay. Moving on to the next slide. Okay, oligosaccharides or polysaccharides. When more than two, when more than two, it means three to seven monomeric units are linked together. It's form oligosaccharide, and more than 7 units are linked together. They form polysaccharides. Large linear branched molecules can be made from simple repeating units of. Okay, when it may be like, consider the example, many glucose molecules could be linked in the same way, that is unbranched. When one glucose is linked, other glucose is linked, but the second glucose bind this link and the branch, that is when state is going as some branching is occurred, like in the tree, this is branching, so it may form unbranched and branched polysaccharides. So when many monomeric units like 1, 2, 3, these are 3 monomeric units are linked together. But see here, as many monomeric units are linked together, but there is a branching. It's a straight monomeric unit, but there is a branching at the heart. How these are linked? These have alpha and beta linkages. So this is here I have drawn the structure of starch, a polysaccharide, which is also present in potato, and we know that potato are sweet in nature. So, but sweetness is lost, okay, with the passage of time and something like that. It's actually the starch that is present which is in potato. Okay, the starch, for example, is a polysaccharide made entirely of glucose units joined together, but they are branched. Glycogen, for example, is a polysaccharide made entirely of glucose units joined together. So, starch and glycogen, we know that excess glucose is stored in the form of glycogen in animals. And in case of plants, in starch, it is stored in the form of starch and cellulose. So short chains are called oligosaccharides and long chains are called polysaccharides. Okay, moving on to the next slide. Complex oligosaccharide. Okay, other than monomeric unit of monosaccharide, other groups are attached. That is amine groups. That is N-acetylamine group. These groups are attached that form complex. In many cases, the sugar sequence is non-repetitive, so there may be different sugars. Okay, number one, many different molecules are possible, such as complex molecules are usually linked to proteins or lipids, as in the molecular, which is a part of cell surface. As I told you, proteins are remaining in the cell surface membrane, and then there are certain carbohydrates that are attached to that proteins that give the function of reception. So these are the complex molecules comprised present here. The partial molecule that defines a particular blood group. In case of blood group, that is the complex molecular is one of the examples. In blood group, we have specific ABO antigen which has a specific carbohydrate group. So that's complex molecular. Moving on to the next slide. Okay. Here I'm going to sum up all the things we have discussed about what is carbohydrate, what is the general formula, what are the different types of carbohydrates. Now, what are the functions? Here I have given functions. I think it may be equivalent to 20, but I think these functions could be classified into different categories. Later on, I'm going to give you a segment. So, carefully listen the functions of carbohydrate. Carbohydrate provide energy and regulation of blood glucose level. This is a common function that everybody knows. It will prevent the degradation of skeletal muscle and other tissues such as the heart and liver and kidneys. So we know that carbohydrates are essential component of our cell. So that's why it's necessary to maintain that structures. It prevents the breakdown of proteins for energy. Okay, we know that our body consists of 70% of protein mass. So in case mostly the people prefer to when they are going to the dieting phase, they less prefer the bread and that carbohydrated content. When this content is not available, actually the body breaks down proteins or fats. So this is the thing how the obesity may be reduced. Though I am working on it, let it move on. So it's a prevent degradation of those proteins which are present in our body if carbohydrates are present in our body. So moving on to the next function, carbohydrates also help with fat metabolism. If the body has enough energy for its immediate needs, it stores extra energy as fat. So I think I don't need to explain, when you consume extra carbohydrates, you actually gain the fats because that energy stored as fat. Carbohydrates are important and component of many industries like textile, paper, lacquers, and breweries. Detoxification of physiological importance is carried out to some extent with carbohydrate derivatives or detoxification. Okay, detoxification is basically toxic nutrients or toxic metabolites present in our body are somehow basically important in physiology of the cell. Agar is the polysaccharide used in culture media like gelatin or food. Agar actually is a jelly-like substance which is actually used for culturing microbacteria. So that is one of the functions that carbohydrates have. Carbohydrates form a part of genetic material like DNA. I told you that basically ribose or deoxyribose sugar are part of DNA. In case of DNA, deoxyribose sugar. In case of RNA, ribose sugar in the form of deoxyribose and ribose sugars. Moving on to the next slide, that is also about the functions of the carbohydrates. Hydrochloric acid found in between the joints acts as synovial fluid and provides frictionless movement. So basically, these are the joints. We have joints in our body where the derivative of sugar. That's why I have to mention about the derivatives in my slide. So these derivatives actually perform the fluidity such that there is less friction, the less wear and tear between the joints. They help make up the body mass by including all the parts of the cells and tissue. We know that that's a simple. Our body is made of fats, protein, lipids, water. So that's the major content. Adequate storage of hepatic glycogen helps in detoxifying the liver. So it helps in the detoxification of the liver. The next section, they form components of biomolecules which have key role in blood clotting, immunity, and fertilization. Blood clotting, immunity, and fertilization. Immunity is basically, we know that a defense system of body, and fertilization is a mechanism which sperm and egg meet and result in the formation of a new embryo. And blood clotting, we know that as we have cut on the body, blood is released and our proteins are activated, our carbohydrates and certain enzymes are activated to stop that flow of blood and results in the formation of clot. Carbohydrate is basically the main fiber of diet and provide the bulk fiber for blood digestion. So we know that fiber form the roughage in our stomach, and that is good for basically digestion and movement, good movement for our bowel, so that we have proper working intestines. Carbohydrate help clear gut and prevent constipation. Okay, starch is a form of food that is stored in plant. I told you about the potatoes. It provides sweetness to the food, as it is named from sweet. Pectin and hemicellulose are the structural carbohydrate plant cell walls. Okay, I have told you about that. It plays important roles in the cellular organization process. In case of cell membrane, they form the receptors. Cytokine form the cell wall of fungi and outer skeleton of the insects. So that is the major function. Urine is structural carbohydrate present in the bacterial cell wall. This function could be mentioned in this difference could be mentioned in relation to prokaryotic and eukaryotic cell. So these are the functions of prokaryotic and eukaryotic cell. So these are the functions of carbohydrates. I have discussed in detail. Moving on to the next slide. Okay, I have mentioned variety of functions here. So you need to classify in three categories of different carbohydrates. That's your task. And I am going to leave this presentation in the form of PDF in my YouTube link, so you can have from this presentation from there, and you can also subscribe my way or like my page to have that presentation too. Okay. Okay. Moving on to the next slide. Thank you so much. If you like the video, do comment, subscribe, or share, and you could have this presentation in PDF format in the link below, and you can find this presentation on my page, Incredible Wow. Thank you so much.
Thank you. Navana. Ay, ah. Time भी लगेगी